Our Friday Conservation Sciences seminar series. So today we have the pleasure of hosting Dr. JJ Apodaca. So JJ he's a colleague and friend of mine. I guess for about 15 years we've known each other now or so. He comes from the salamander world, works on salamander genetics, in particular conservation genetics, some systematic issues, species delimitation issues as well. And I thought JJ would be a particularly interesting speaker because he kind of took the normal academic track of getting a PhD. And then he had a position in academia and he decided to move off into the world of like being a little bit less academic. It's still you do a lot of academic work, but more applied. And so he started a nonprofit conservation agency called Tangled Bank Conservation. And he's also the executive director, remind me what for the Amphibian Reptile Conservation Network. Is that what it is?
Amphibian Reptile Conservancy, ARC. Yeah.
Conservancy. So, so I think JJ, he's going to talk a little bit, about his research and his path here today. And I thought he'd be an interesting speaker to have, especially for our graduate students. For a lot of them, you guys don't go into academia, and so I think JJ will be, kind of an interesting example of creative things that you can do with your degree, your PhD in conservation sciences. So with that, JJ, we're glad to host you and have you today. I'll let you take it away.
Cool. Thanks so much, Ken. Yeah, let me try to pull this off here without messing it up. How's that?
Yeah, looks good. Perfect.
Okay. Cool. Yeah. So thanks again. I should say Dr. Kozak for inviting me and for all you for having me. It's really always an honor to get invited to seminars in general. I think it's really cool, but it's really extra special for me when it's something like this. It's directly focused on conservation. And I think, Dr. Kozak asked me here today, as he said, because I feel a bit of a strange niche in the world of ecology and evolution and conservation biology. And it's a niche that I really relish and enjoy and feel very lucky to do on a day-to-day basis. And that's that I get to be kind of a bridge position between, really pure science and pure wildlife or habitat management. And I do this in another really strange niche, which is that, as a lot of people like to call it, as Ken said, a kind of an alternative career path, which I'll talk more about later and define more clearly. And I'll just say that I increasingly get asked to do these talks on alternative career paths, which is pretty cool. And I'm just so busy that I've started to say no a lot, which is something I used to never do. But again, Dr. Kozak really convinced me that for this one, by selling me on how interested you all are on applied conservation, and I really appreciate that. And so here I am, trying to get this to work. There we go. All right, so everybody loves starting a talk with a definition, right? And I know you all will be especially in love with me defining conservation biology, since this is a conservation biology seminar. But bear with me here. I have a point, and it kind of shows you how I think about conservation and some of the problems that I've tried to address in my career. And that's really that if you like Google conservation biology or look in a textbook, you get something like this, right? The study of attempts to protect and preserve biodiversity. And then it might mention something about how it focuses on both biological and social factors. Now this sounds about right? Nobody really thinks too much about the definition of the field they're in. And there doesn't seem to be anything terribly offensive here, but I just can't tell you how bad I think this kind of definition is. And I'm not just saying that because I'm a grumpy old man. That might be true too. But the reason that I think it's not just wrong, but it's also really harmed the field of conservation biology is that it's conservation biology is such an applied science, right? Which you actually see in all the textbooks. It's an applied science, but and there's so many other applied sciences out there. So I always like to look at this definition and apply it to another applied definition and see how it sounds. And that's, here's, there we go. So let's take aeronautics, for example. One could argue that aeronautics is a pretty damn important applied science, right? So if we apply a similar definition here, we get a pretty silly sounding field. It focuses on on the study of attempts to fly through the air, both the physics and the social factors, right? Which is really silly, right? That's not how they define aeronautics. And if this is how we had approached the field in the beginning, we wouldn't have flights today. In fact, most people forget that Orville and Wilbur Wright were not supposed to be the first people that achieved flight. There was a very well-funded scientist who everybody was kind of backing and had tons of theoretical papers and all that sort of thing. And so the point here is that if you sort of look at the true definition of aeronautics, the science and or practice of travel through the air, this is more of like an applied definition or applied science, right? And so let's see, here we are. So I like to think of conservation more like this, right? And I've kind of come in the last couple of years to think of it more like this, that it's a science of uncertainty and an art of probability. And I completely stole this idea from another very applied field, which is medicine. So this is what's known as the Osler principle in medicine. But I think it really applies well to conservation biology. And it makes another really good point here is, if all we do in conservation is work on the science side of things, that things never get better and populations never recover, and we continually lose species and habitats and ecosystems. And so I always, compare it to, it's like the equivalent of learning all we can about medical issues, but then never doing anything about it. And, it'd be like going to your doctor and your doctor saying, "Oh, I'm sorry, I know exactly why your heart is failing, but we can't do anything about it because this is science after all." So what I try to do in my work is to bridge these two sides together, to do the science and the art of probability, to lower that uncertainty. That's where the science comes in, obviously, but also to get conservation programs started that have at least some probability of working, right? And we're going to fail, but that's important. And in my opinion, for all you that are looking for a career in the conservation world, we need a lot more of this art of probability side. In other words, we need more people taking that science and translating it and doing something with it. And so that's kind of my soapbox on this. And I also say that I never used to have a picture to go here in this talk or when I talked about this. And then along came DALL-E, which, if you've been living in a cave, is this awesome program that takes your prompts, such as a conservation biologist who is also an artist, and makes a picture using AI. It's really, really a remarkable technology. And this is my favorite one that it created for those prompts. So there you have it, a conservation biologist who's also an artist, according to AI. I'm just going to imagine you're all laughing here. And so that's where I really decided to focus my career on, not on AI art, but on applied conservation. I had, as kind of mentioned earlier, I'd taken the classic path in academia. I'd done a postdoc in a really good lab, and I'd gotten, some publications and eventually got a job at a small school where I thought I'd be able to really focus on applied conservation and research. And, the unfortunate thing is that it turns out that when the place hires you to be a faculty member, they actually want you to do very faculty member types of things, which is bad, was bad for me, I should say. Like they want you to go to faculty meetings and advise freshmen, which I found to be, both a very special kind of torture. I didn't really want to do those types of things. And at the same time, they're expecting you to bring in grant money. So, what that really turns into is you're bringing in grant money to hire a postdoc to do all the stuff that you really want to do, or at least I really wanted to do. And so that you can do these other types of things that I didn't really want to do. So anyway, long story short, after a few years, I decided, I really wanted to jump and focus on that applied conservation biology side. And at the time I had a few contracts, and I took those and I built a genetics lab and I started a business, which is Tangle Bank Conservation. And it really took me a couple of months to realize how weird this was, and how unlikely this was. But after trying to explain it to a bunch of people, like what I was doing, it really kind of sunk in that it was something different. And so as I've tried to explain it to people and it becomes really difficult, I figured I would employ Dolly here too to come up with a graphical depiction. So this is Dolly's version of what an alternative career path looks like in conservation, which I just love. And it really demonstrates that even AI understands what I do for a living more than my family does, which is great. Okay, so I'm not going to do AI art throughout the talk if you're worried. So that brings up the question of what do we actually do? What do we at TBC? And I like to tell people that, I've built this business on the concept of capacity. So all throughout the 2 decades that I've been involved in conservation, which is a little hard to say now, the number one thing that's needed has been capacity. If you talk to an agency or an NGO employee, they always say, we need more capacity to be able to accomplish on the ground conservation. And so I thought, well, we can be that capacity. I can build this business and be that capacity. And it's also based on the idea of recovery. And the best way to describe this is if you look at a very basic outline for what's needed for species conservation. I'll throw that up here. There's this glaring hole in the recovery side of things. So research is very well covered by academia. Regulations is the arena of the agencies. Conserving land and habitat is really covered by land trusts really well. Advocacy is pretty well covered by NGOs. But nobody is doing a really robust effort to recover species. Now, that being said, there's a lot of groups out there that are attempting this. Federal agencies do this to some degree, and state agencies, but most of them are overwhelmed, and it's not their primary focus, and it can't be their primary focus for various reasons. And so again, that's where I pictured Tangle Bank coming in and trying to really start that recovery process. And as you'll see later, this is where a lot of where ARC, Amphibian and Reptile Conservancy, that I also run, comes into play as well. And so very fortunately for me, when I was, starting Tangle Bank Conservation, Fish and Wildlife Service also revamped their way that they looked at species recovery. And this happened via 2016 Directorate's mandate. And for the first time, the U.S. Fish and Wildlife Service had a legal mandate that explicitly focused on genetic factors, which was pretty lucky for me as a geneticist. And this is, generally called the 3 Rs. A lot of you have probably heard of it at some level, or hopefully have heard of it. And it's really part of their species status assessment, or SSA, process. And it's a really important part now. And it's, worked its way into those SSAs and recovery plans and all these things that are important for endangered or petitioned species. And so a lot of people have heard of 3 Rs or might know what they are: redundancy, representation, resiliency. But real quickly, because they're so important to the rest of the talk here, I'm going to kind of briefly go into them and describe them. So the first here is representation, which basically says we want more historical and genetic diversity. And the idea is that you want to preserve as much historical diversity as you can, which gives a species a better chance of surviving catastrophic change, like climate change or major disease. So in other words, this is like conserving phylogenetic patterns of a species, which is pretty awesome. Like, when Ken and I started in this field, there was no legal thing that even kind of considered this. So it's kind of neat to see it come into a legal sort of framework. And the second one is redundancy. which is basically within each of those lineages, more populations are better than less, right? Pretty obvious here, helps protect against catastrophic events and stochastic processes, but again, extremely important here. And the last is resiliency. And so what that says is that large populations are better than small ones. Again, pretty obvious one, but an important implication here is that we aren't just talking about the size of the population, but at least in my opinion, but also the amount of genetic diversity within each population. So again, large, more genetically diverse populations are much better than small and less diverse populations. So again, those are the 3 R's. And now I'm going to kind of go into what we do at Tangle Bank and how we fit into the 3 R's process and provide that capacity that I was talking about. And so, again, I'll throw a couple case studies up here. Let me just say that generally we at Tangle Bank Conservation do between 40 and 50 projects a year. So I'm just picking a small selection of some of my favorites. We do tons of really fun, cool stuff. But I think I'm going to start with a fish and a really cool genetic tool called eDNA. Again, I'm assuming most of you have heard about it, eDNA, but if you haven't, this is some really awesome CSI stuff here. I've been doing eDNA almost since the beginning, and I'm still pretty amazed at what we can do in terms of picking up DNA present in the water from shed skin or whatever it might be. It's really a wild idea, and every time we test something new and it works, I'm just blown away, including, things like getting DNA from dirt or swabs from a cover board, or there's just a million things you can do with this, but really cool. So how does this tie in? Well, you can't really manage the redundancy in a species, right? You can't really make those decisions to recover a species and recover or create more redundancy, or even assess if a species does or doesn't have redundancy if you don't know where populations are. So we actually use eDNA for this purpose a lot. And one of our coolest examples here is this cool fish, which is a herp guy. This kind of hurts me to throw a fish up first, but I'll get to a lot of herps. But this is the sickle-fin red horse, which is a Moxostoma or a sucker species. A lot of you are probably familiar with, the normal, where there's actually a lot of species of suckers and red horse in the United States, but there's generally just red horse in a lot of places. This guy is its own species, found in a very small range in Western North Carolina and Northern Georgia. And it's actually an undescribed species because the guy who's worked on it for a super long time and actually discovered it has really just sat on the description for about 30 years, which is sometimes just how it goes in science. But anyway, it's still been recognized for centuries as a distinct species, even by the Cherokee, and who actually have a cool name for it called Jungitla, which means feather and cap for that cool dorsal fin that they have. And it's actually a really culturally important species for the Cherokee. However, with the damming of many of the rivers in their home range here, the species has nearly disappeared. So we've been working with the Eastern Band of Cherokee, the U.S. Fish and Wildlife Service, North Carolina, state of North Carolina, state of Georgia, and a whole bunch of other partners to help bring this species back. So we've developed an eDNA protocol and sampled all throughout their known range, or at least historically known range. Obviously, again, that's important for the redundancy to find any known populations, anywhere they might be. We also, getting outside of the redundancy part, we did a full mitochondrial next-gen sequencing to sort of identify lineages and assess taxonomy and genetic diversity for this species. And really the entire team here is working on doing reintroductions, which is really cool. So this is taking like the basic data and going out and being able to put back thousands of sickle fins that have been raised in U.S. Fish and Wildlife Service hatcheries and putting them back into native waters every year, thousands every year, which is really cool. And based on our genetic work where they're putting them, and eDNA work where they're trying to stitch populations back together. Also using those eDNA results to guide the restoration of that native habitat conditions, removing dams and doing that sort of work. So that hopefully one day the species is once again like a really major component of Southern Appalachian ecosystems. So just a really neat project and fish species there. Next, we'll jump to resiliency, which again, growing population sizes. Of course, in the sickle fin, we're doing that too with reintroductions, but we'll move on to one of my favorite species, which is the bog turtle, which is just such a really cool little turtle, our smallest turtle species in North America. And this is a species we do a ton of work on. And we just recently got a really large multi-state wildlife grant, or AKA a CSWIG, to work on the southern range of this species. This is one of the most, if not the most, endangered turtle in the United States, and they face all kinds of threats. They're really, really in trouble. But ultimately, like what we really need are more and larger populations of these guys. There's a lot, not a lot, but there's a good number of populations, but a lot of those populations are a couple individuals, especially a couple old individuals. So we've been going out for several years now and restoring habitat, caging nests, and head starting these guys. So every year we put 6 full-time people in the field for about 2 months to trap females, and fit them with radio trackers, and then they follow them around during nesting season, find where they lay their nests and cage those nests. It's a ton of work, but it really pays off with some of the really freaking cutest turtles in the world here. If you see these guys, which are just incredibly awesome little turtles. We're also using eDNA and on the ground surveys to find new sites and restore them, which is it's a bunch of work. Again, but really worth it. And this is just some drone photography here that I took that on the before and after for a bog. So the before is on the left, really grown over, which is not good for these bog systems. Huge amounts of woody vegetation. And so we generally go out and restore between 5 and 10 bogs a year. And they start out like this on the left, super overgrown. We cut out all that woody vegetation that's crowded out the bog and sucks the water out and all that sort of stuff. And it really restores this foraging, breeding, and nesting habitat for the species, which is a incredibly important thing to do for these guys. Also, that type of habitat is one of the rarest, if not the rarest, and most declining habitat types in the Southern Appalachians. So helps out a lot of other things, including, a lot of super endangered plants like this listed pitcher plant and sundew and all kinds of those things. So really, really cool work there. All right, so talking next about representation. And so again, this is like, sort of the classic phylogenetic question of what across this species do we need to conserve in terms of historic phylogeographic lineages. And so I'll start, in my opinion, with like the greatest phylogenetic puzzle out there, taxonomic puzzle out there. Maybe I'm just saying that as a herp guy, but that's pine snakes. And so this is a system that has really vexed herpetologists since the beginning. They're super morphologically variable. Early mitochondrial sequencing didn't help. They didn't really show any patterns or clear anything up. They integrate a lot. And on top of that, they're just super rare, basically. So fossorial, very hard to sample, very hard to get samples from, except by pure luck, basically stumbling on a dead one on the road, or ideally a live one on a road. So in general, they're just a huge mess. However, obviously, of conservation concern, and the Florida subspecies was petitioned a few years ago. So Fish and Wildlife Service had to figure out what to do with this species because it was only the Florida subpopulation was petitioned, which means that that was all they could look at, and they had no idea where the subspecies ended or whether it was even valid. So before they could move forward and take any kind of conservation action for the species, they had to kind of answer that question. And so we came in and did a full phylogenetic and population genetic assessment of the species. By some miracle of networking, we ended up with this species getting over 480 samples. We're actually well above 500 now, which again, if you've ever come across pine snakes or dealt with pine snakes, that would be pretty amazing. And we did a bunch of ultra conserved elements sequencing. We did 3RAD, which is a restriction type of next gen sequencing, and yeah, did phylogenetic and population genetic analyses, and found out that indeed they actually are a mess. But you can make out some valid patterns here. We can find some pretty cool patterns that show sort of the historic phylogenetic history here and current population genetic history. And so basically what we found and what we're able to show them is that kind of this olive green here can be reasonably considered the Florida subspecies. And so this is a species that they haven't made a decision on yet, but they now have our recommendation on this, and the SSA team is moving forward. So we'll see what they do with that and what kind of decision they make with the genetic data. Another cool example of work we do with this, with representation, is with the alligator snapping turtle, which is, another petitioned species that was under review. And, we go from the smallest turtle in the United States, the bog turtle, to the largest freshwater turtle in the United States, the alligator snapping turtle. Super, just an amazing species, a really cool species. But this was also another species like the pine snakes that had a lot of taxonomic uncertainty. It had originally, a few years ago, 2 lineages had been described out as new species, and then one of those was sunk, and there was rumors of another. So really, to move forward, Fish and Wildlife Service had to know the lay of the land with this species before they could make any kind of conservation decision. And so we did, very much the same thing. We went out, got a bunch of samples across the range, over 400 samples. We're now again up to almost 600 for the species. A huge sequencing effort for this guy. Very quickly, in less than 6 months, we got all these samples and read, sequenced over 5 million reads per individual, and just got a huge number of single nucleotide polymorphisms across the range of this, which was really cool. And it showed these really strong patterns for the species that basically told us that each drainage for these guys represents a pretty substantial evolutionary lineage. And so what that looks like is this, like, these are the kind of evolutionary lineage drainages that we gave to Fish and Wildlife Service. And they actually recently decided that because of this number of historical lineages and specifically the lack of redundancy within each lineage, the species should be listed as threatened. So that's not official yet, but they've recommended it. Great conservation win for this species, I think. And this wouldn't have happened if all these lineages were considered together, right? If we didn't have that evolutionary history across that they were legally mandated to kind of protect, this would have been a no listing decision, which was actually where it was headed before our genetic data came in, which was this is pretty cool. Another awesome side effect of this data is that with that much data, we can identify exactly the exact river where poached individuals came from. And so last year, well, a couple of years ago, there was a collection of about 60 adult alligator snapping turtles. We used this data to sequence all those individuals and put them back in the exact river where they came from. It also helped prosecute the poachers who were telling the agents that they didn't cross state lines. We were able to show that wasn't true. So really cool, got prosecution there, and more importantly, got the over 50 of those adults, some of them well over 100 pounds, back into their home water. So that's really, really important for those guys. If you know anything about turtle biology, getting the adults back is like putting thousands of juveniles back. And this is actually, that kind of work is actually one of the major avenues that we're doing right now within Tanglebank. The illegal turtle trade is an enormous problem that I could have an entire talk on, because it's something we're working a lot on right now, and agencies are working a lot on. There's a big movement here because it's just been realized like how many millions of individuals are being trafficked out of the country. So we're actually right now building genetic databases for several of the most poached species, and working with Fish and Wildlife Service. And so they come to us when they get a confiscation, we sequence them and get those adults back in the wild, and which is a really, really rewarding thing to be working on, and really a lot of fun. Okay, so that's Tanglebank Conservation, and just like a little taste of what we do and what I've been working on, with kind of going out on my own. But after, a few years after I started Tanglebank, I was asked to take over as executive director of the Amphibian and Reptile Conservancy, which is a nonprofit dedicated to conserving herpetofauna and their habitats, if that wasn't, really clear from the name. And so this really kind of continued this alternative career path in a new way. I'd been involved with ARC and their project Park for a long time. A lot of you might be familiar with PARC Park, which is the kind of expert network that's fiscally sponsored by ARC. It's a project of ARC. And so, this was really important in my career coming up, and being involved in Park, and then later on in ARC. And so this was really kind of a dream position, mainly because I could really develop my vision of that on the ground herp conservation that I thought was so important. Again, kind of goes back to my original version that I was talking about there about recovery, and so I couldn't really say no to that, and that's how I found myself running 2 organizations and being chronically stressed, but also very fulfilled. So what do we do at ARC? So at ARC, we focus on PARCAs, or what are known as PARCAs, PARCA, priority amphibian and reptile conservation areas. So some of you might be familiar with like important bird areas. And this is, sort of our herp version of this, of trying to find the most important areas across the country, and getting conservation started on those. So at ARC, we identified those PARCAs across the country. We go in, we do inventory and monitoring and science, and we create conservation actions, and we really implement those programs on the ground. So I like to think of us as like a catalyst for conservation. And I think this is so incredibly important. And another missing element in conservation is that having a group or some people that will drive conservation forward, and that will just push. There's a lot of people interested, in a lot of agencies and stuff interested in conservation. And so if you can have somebody who's willing to organize and push forward and get get some skin in the game, you can get a lot done, which is what we try to do. So this is what our current distribution of PARCAs looks like, and this is we're exactly at half of the country right now, although there's no native terrestrial herps anyway in Hawaii, so we're a little bit over half. And you can see these are pretty big landscapes, and that was on purpose. Again, we can talk about that later, but as compared to IBAs, important bird areas that can be as small as a single wetland, to really recover herps, we're talking about substantial metapopulations across the landscape. And so that's how we design these. That's how I envision parkas, and that's how we've been implementing them. And the other thing that this does is it allows us to really partner with a lot of organizations in these areas and again, kind of congeal that conservation and get something going. This year we'll be identifying a bunch of new ones thanks to some funded grants. And in the next few years, hopefully we'll have all of them done across the country. And so this is pretty exciting for me because I've actually been working on identifying parkas for just about a decade now. So pretty neat to see the end in sight. But really why I'm most excited about them is that we're taking these parkas, these priority amphibian and reptile conservation areas, and implementing them and creating long-term on-the-ground programs. So in other words, we're taking those, our team is looking at like, where are the most important parkas, where are those most important priority areas, and where can we make a difference? And to be cliche, where can we move the needle on conservation? And so we've got a lot of really cool projects, I think, going on for this. One example here is our Gila program in New Mexico. This has 3 listed species, federally listed species, and a lot more at-risk species and species that are in trouble. But the 3 listed species are the Chiricahua leopard frog, the narrow-headed garter snake, northern Mexican garter snake, which are these 3 species over here. And so really for this program, this is a 10 to 20 year commitment from ARC. And in some cases our partners to go in. We're going in, we're doing inventory and monitoring, we're doing invasive species management, lots of problems with bullfrogs and crayfish and some grasses out there. We're doing habitat restoration and creation. The hydrology has been really messed up out there for a long time. And so we're working to fix that. We're working to take out non-native fish and put back native fish for the primarily for the garter snakes. We're doing population reintroductions and augmentations, and ultimately across the entire landscape, we're rebuilding these healthy metapopulations that these things need again. And that's going to take, a long time. It's going to take 10 to 20 years if we're lucky. And so we're doing that with just a ton of partners. So I just really show you this because it's important in this level of conservation to think about who you can bring in, and especially for these long-term projects, what those look like, and really focusing those resources for purpose, for strategy, and for long-term conservation. Another awesome project that I'm really excited about that we're doing is one in the Bankhead, what we call the Bankhead Parka, which is in Alabama, kind of north western Alabama. And again, 2 federally listed species here, the flattened musk turtle and the black warrior water dog. And for this project, we're doing a really cool approach where we're taking eDNA metabarcoding, so sequencing everything that's there, because in addition to these 2 listed herps, there's also a bunch of listed fish and a bunch of listed mussels and snails. And we're combining all that data with threat mapping, what I call threat mapping done by citizen scientists. This is where people are going out, they're canoeing, they're kayaking, and they're identifying all these threats to the species, from low-head dams, hanging culverts, areas where high sedimentation is coming in. This is an area that used to be really heavy with mines, so there's still like open mine pollution sources running in. And so we have funds from NIFWF, National Fish and Wildlife Federation, to remove those barriers to gene flow and to fix those problems of pollution. And so what we're doing is we're creating maps right now where we're combining the eDNA and those threat maps. And, from that, we can sort of prioritize where to go in and spend these funds and fix these problems. It's really, really cool approach. And again, a ton of partners here, as well. Again, NIFWIF has funded a lot of this, Forest Service, Fish and Wildlife Service. So just a cool, again, long-term 10 to 20 years project that we're focusing on. And this is kind of our norm. This is what we're going to be doing for the long term. So if you're really into herps and herp conservation, watch out for our programs starting in, or either going now or starting in 2023. We have 19 of these areas where we've committed to long-term, large scale, landscape scale restoration and those sorts of things, projects across the country. And we're constantly looking for new funds and that sort of stuff to do this. So really, really excited to be able to build sort of this level of program and these types of conservation programs across the country. It's super cool. So if you want to keep up with what ARC is doing, please check out our website, which is pretty new, arcprotects.org. And it really helps us out if you sign up for a newsletter, and heck, if you want to go buy something too, that'd be great. But just keeping up with us and showing support is a great way to support what we do. So I want to leave a lot of time for questions and that sort of stuff. So and everybody loves a shorter seminar anyway. So with that, I'll just kind of say thanks. Obviously, I don't do all these projects by myself, or far from it. So I've got a lot of great people working with me and for me, and a lot of great funding sources.
All right. Well, excellent. Thanks for a really great talk, JJ. I'm sure JJ is obviously happy to take questions right now. So you can either unmute yourself if you'd like, or throw them in the chat. So I guess I'll get it started here a little bit. Can you talk a little bit about, so you did these things, discuss how did you actually go from being a professor to starting a nonprofit? You talk a little bit about what the hurdles were and kind of the connections that you needed to make, because that's a really interesting transition that I think people would be interested to hear the details about that, how that came about.
Yeah, sure. Well, man, I don't know where to start there. I guess the place to start would be that I was very fortunate and had been involved in the conservation world for a long time. And so I was bringing in contracts. I knew what was available, and I had made a lot of contacts. So I didn't just one day go out and start to sort of hang up a shingle and say, I'm open to take your contracts. But I had a lot of those built up, and luckily I kind of saw the end coming, with academia for a while. And so I actually opened Tangle Bank a good amount of time before I left academia and started getting contracts and doing contracts on the side, and was able to buy a lot of equipment that way and that sort of stuff. And so yeah, I mean, that's a gross oversimplification of it, but
right. So you run it entirely on contracts, right? At this point, the whole agency pretty much, right?
Yeah. Well, Tangle Bank is actually not a nonprofit. Tangle Bank is a company.
Okay.
ARC is a nonprofit, but but we're entirely on contract. There's, like I said, there's such a need for capacity out there. I mean, we've had contracts doing everything from, I focused on genetic work here and some of our field stuff, but we've had contracts from the Nature Conservancy building wetlands, from NRCS doing GIS analyses, from there's tons of that. But but yeah, the way I was able to do that was by being very involved in the conservation community and networks for a long time, and having a lot of connections. You're not going to show up one day and say, hey, I can do a project, and have people give you a contract.
Gotcha. Got some questions come up here in the chat. I don't know if you can see those or not.
Yeah. Okay. So, Colleen, with eDNA, how do you know if you're getting multiple samples from one individual? Well, I don't know what you mean by that. I don't know if you mean a single site or across sites. Within a single site, you really can't tell if you have one individual or 100 individuals. There are some general trends, like if you look at a bunch of samples, generally sites with more DNA tend to have more individuals, and vice versa. But you could be 10 feet away from a dead individual and there'd be one individual, or you could be 100 feet away from 20 live individuals and you probably get more with the one dead. So it's not perfect. As far as like in a river setting, if you're talking about multiple samples or one individual, well, we know, and we've tested, and other folks have tested kind of how far those samples go, and it tends to not be that far. Like we tend to only get DNA from hellbenders, for example, at just under 1 kilometer away, and then the signal kind of disappears. So generally, that's kind of my rule. If you're within 1 kilometer, you might be detecting a similar population, but apart from that, you're probably not. Does that sort of answer that question?
Yeah, this is Colleen. Hi. I was wondering about how when you're pulling these genetic samples and putting them into trees, how you know if you're like overrepresenting or underrepresenting a particular part of it because you're getting a ton of DNA from one individual, or if you know if you're like spreading it out to different individuals or how that.
Yeah, I think maybe I made it confusing that we're not using the eDNA to build phylogenetic trees or to do any of that sort of stuff. The eDNA is simply like a 1 or 0. It's they're there or they aren't there. Yeah.
Got it. Okay, so like for the alligator snapper drainages, those were from actual individuals.
Yeah, those were all samples.
Well, that makes sense. Okay.
Yeah.
That's interesting. I haven't heard much about eDNA. That's neat. Thank you.
Yeah. I guess next we have Julia, who said no parkas in Minnesota and Wisconsin, no partners. There's just not important herps in Minnesota or Wisconsin.
Story of my life.
Yeah. No, it's just not a state that we've gotten to yet. We're getting there. The parka story has been one of getting patches of funding, and so the upper Midwest has not come through for that. And like some of the Midwestern states that got funded because we tacked it on to a monarch butterfly initiative thing. And so we're getting to Minnesota and Wisconsin. It's just hasn't happened yet. And next one is, how did you make the hop from eastern regions to western, any Midwestern in between? Was under the impression that as you move west, you lose salamander species and other herps. Yeah, I mean, so when we look at the most important, highest richness, most number of imperiled species, that sort of thing, there's really two huge hotspots, which is the southeast and the desert southwest, or the different parts of the southwest. And so that's basically why you lose salamander species as you head west, but you get way more snakes and lizards and that sort of stuff. So again, just we make most of these decisions based on data from richness or species need or that sort sort of thing. I don't know if you wanted to clarify any of that, Madeline, on any part of the question I didn't address adequately. No, okay. Can I briefly describe how experiences differ between working with a company and an NGO? Yeah, there's a ton of differences. One, when I started my company, and to be clear, I'm an LLC, a single operator, or single owner. I've got a lot of employees, but there's challenges to both, and advantages and disadvantages to both. Working as an LLC, you're basically completely your own boss, which is great, except for obviously answering to funding. There are a lot more funding opportunities that are open to nonprofits over companies. That can become an issue. But for the most part, you have a lot more flexibility with your own company. Working as an NGO, again, there's more funding opportunities open, but you've got this other level of fun, which is a board, right? So you always have to deal with a board, and a board is made up of a lot of individuals that may or may not agree with you, and they may or may not agree with your vision of what you need or where you need to go. I'm very lucky. I have a great board and a great working relationship with them, but that's one issue of it. I think also, working for an NGO, sort of people don't question your motives as much. At least early on, people were questioning why I was doing a for-profit sort of thing, which, like, as if profit is a really bad thing, or making a living wage is a bad thing, sort of thing. So yeah, that's just another side thing, but I think that's just a lot of people think nonprofit is no money or low money, and in fact that could be the opposite. You have a lot of these, a lot of the bigger and nonprofits and NGOs have executives and upper management with really huge salaries. So this is a good one. The next one: how do you balance the potential benefit of parkas by improving habitat for many very at-risk species, and the potential devastating consequences of increasing the profile of specific locations on the ground where poachers can go to illegally collect rare species for the black market? Is that a concern? If so, how are you mitigating that? Yeah, that's always a huge concern for us. And not just poachers. Herps tend to get loved to death. You go out and people go out and they flip the same rocks and logs, and it ruins habitats and kills things. And we've seen hellbenders overloved to death because everybody wants to go see one, and they'll go, stress them out until they die or whatever it is. In general, parkas are very large. So we do have some small ones, and we tend to hide those because of that risk. So, sort of where I draw the line is like, if somebody can get the information that we're giving out from a field guide, I don't worry too much about it. And that's kind of where we stand. On the other side, I'm hopeful that as recreational herping and everybody on Instagram and stuff going out and herping gets more popular, and poaching is much of a concern, that those communities start sort of policing themselves and doing that. But that might be a ways off. So the other way that we mitigate that is that we work a lot with law enforcement. I think most wildlife law enforcement that you talk to now will tell you the same thing, that when they got into the field, they were, dealing with thought they were going to deal with deer and turkey poaching, and now pretty much all they do is non-game, and or a big concern. So they're learning. We're doing a lot to educate them, especially in the turtle world and the turtle poaching world. And they're great about it. When we have workshops all the time, we, in-person meetings with state law enforcement. And so that's, one of the major ways that we're mitigating that. All right. So Jaron, what types of careers are out there for conservation biologists and some of the programs or companies that you talked about? What would be your advice for folks with graduate degrees looking to work in conservation outside of an academic or agency setting? It's just so incredibly varied. And so I think the important thing is that you sort of find your niche and your passion and what you can really provide to people and be really good at. And that can be all over the place, but what kind of jobs, like the Nature Conservancy always has an incredibly varied list of jobs from state to state. So there's everything from that to, some NGOs have chief conservation scientists now, or, there's just all kinds of stuff. So I think the important thing is to figure out really what you want to do, and like what you're passionate about, and become a resource that somebody outside of academia or agency can value and put to use. Not sure if that makes sense. If you want to unmute or clarify on any of that, or ask any further questions, feel free. But yeah.
It looks like there's one last one there from Alex, I think. And then
okay. Yeah. Wondering how you decide what herps or fish need genetic rescue. I guess by that, do you mean which species to work with initially, or after we've done the analyses? I'll start with the first, like what species to work with. I've got species that we go across the board. Let me just say that from things that I'm like, this needs somebody to work on it, and we'll have to go convince the states for a few years or the feds for a few years, and eventually they'll buy in. Or what drives a lot of things for us is state wildlife action plans, SWAPs. So if you're unfamiliar with your state SWAP, that's something you should get familiar with. These are plans that each state has to write in order to get funded for SWIG grants, for state wildlife grants from the U.S. Fish and Wildlife Service. And those will outline everything that that the state finds or thinks is in need. And it actually lays out a bunch of really great questions for academic work and for students to work on, and that you can get funded. And so those are a great resource. Do you work with state organizations primarily? Again, both federal and state primarily is where we work. I'd say it's a little bit more federal than state, just because they tend to have more money. Has an individual ever come forward to you to discuss genetic work? I'm not sure what you mean by that. I don't know if you're on and you want to clarify what you mean by, has an individual ever come forward to discuss genetic work? Oh, like they wanted to fund it, like an individual? Yeah, actually, we have had a couple cases where just wealthy individuals were interested in certain projects or species or questions, and have funded things, and we've worked with them. In fact, one individual I talked about our genetic databases for turtles. One private individual has basically funded most of the box turtle database, except for the Northeast, which was funded by the RCN up there. So.
Well, it looks like we're actually right on time here. 1 o'clock exactly. So I guess JJ'd probably be able to take one more question if somebody wants to throw one in there. But if not, thanks for a great talk. It was nice to have you and
yeah,
talk about a lot of these things that you have going on, and other opportunities, other ways to kind of be involved. So
yeah, thanks again for having me. It's
yeah. And if if you want to hang out for a minute, I guess other people are probably going to sign out here. Okay, hang out for a minute, and you could chat for a second. I'm just not going to throw you off here or anything like that. But so anyway, everybody's free to go now. Thanks for a great seminar, JJ.